Good morning, everybody. It's 10 a.m. Houston times. Good morning to all MD Anderson family for enjoying science. This is a webinar for all of you from here and from all over the world, all members of our large family. And it's done by wonderful clinicians and scientists from MD Anderson for you scientists, colleagues, and patients. I have to thanks to Dr. Giulio Draeta because he is organizing this large series, and of course to Ellison, Garth, and Liz Ken because they are the engine behind all this very large effort. Without their work, nothing can be done. And of course, I have to thanks to all of you, hundreds and hundreds of attendees who are here present each talk. I have to tell you that we already passed 3,000 attendees for our first 3 weeks, and we are looking forward for you to be with us for the next several months. Also, I have to ask you, please use the chat button in order to ask for questions. And moving forward, it's my pleasure to introduce today Dr. Anil Sood. I know him for a very long time, for over a decade, and I have to tell you that I was all the time impressed by the fact that he is able to combine in a wonderful way the excellent clinical work with outstanding research. If you go over and check Anil's work, he is able to publish extraordinary nicely, both clinical and research. He is a quintessential clinician scientist, basically representing what is the power of our institution. Very strong clinicians doing wonderful research. And with this said, it's my pleasure to let Dr. Anil Sood to run the show. Thank you, Anil, for being here with us, and good luck with your presentation.
George and Alexandra, thank you so much for inviting me and for organizing this seminar series and bringing the MD Anderson community together. I also wanted to just take a second to, or just take a minute to acknowledge Josh Fidler, who unfortunately passed away last week. Josh was the chair of cancer biology when I came to MD Anderson, and it was certainly a home for many physician scientists, including myself, for many, many years. And I just wanted to acknowledge all that he did for me and for many others around the institution and other places. These are my disclosures before I start. So in this talk, I will discuss a little bit about mechanisms of paraneoplastic thrombocytosis, or high platelet counts, and a couple of areas that we've also worked in related to how anti-angiogenesis therapy can impact platelet extravasation and what the biological roles might be, and how platelets can impact tumor cell survival and its implications as well. So, as I entered this field, one of the questions was, why study platelets, especially in the context of cancer? So allow me to just build a bit of background around this. For me, the interest around platelets goes back to my medical school years. This is a picture of Herb Proctor, who was a trauma surgeon at University of North Carolina, where I was a medical student. And he was my advisor and my mentor during medical school. In my third year of school, he went to Maine to give a presentation. He got off the airplane and collapsed at the airport. And he was diagnosed with a pulmonary embolus, or a blood clot in his lungs, and his platelet count was around 700,000. And that was his primary presentation for metastatic colorectal cancer, which spread to the liver. He unfortunately died about a year and a half later, and but it certainly left a huge impact on me in terms of what he went through. And I always wondered, what is it about cancer that can cause high platelet count to occur, and then patients end up suffering from blood clots in the lungs or peripherally. Now, I mentioned Josh earlier. During the when he was in cancer biology, it was clear that you had to know the history behind a field that you were working in. So I always make it a habit to try to learn about the history of a given area. So bear with me just to give you some background. Recognition of platelets goes back a long time. As far back as 1600s and 1700s, people would see small, undefined particles in the blood. At that time, there was really no recognition that platelets existed. And in the 1840s, Alfred Dohn called presence of, or what he termed globulin ductile, which stood for small globules derived from plasma. Max Schultz in 1860s described clumps of irregular shape and different size up to pretty good size, and that size is obviously too large for platelets. And he described these particles are often sharp-cornered or have a granular appearance. Basically, what he was describing was activated platelets, and that's likely why they appeared larger in size. Finally, Giulio Bizzozero in 1880s coined a term that in English is translated into small plates. And he was bold enough to state that there is a third element of blood, that there isn't just red blood cells and white blood cells, but indeed there is a third element that he called small plates. He was actually attacked quite a lot because the dogma at the time was that really there are only two cell types that exist in the blood, that there is no other cell type. Well, he was obviously right that platelets existed. Then the field actually stayed relatively dormant until the early 1900s when scientists both from US and from UK started to work out how platelets originate and recognition of megakaryocytes and so on. So that's just a brief history about platelets. Another very important history component that's important to recognize is Trousseau's syndrome. So Armand Trousseau was a French internist who in 1865 made a statement that if you see a blood clot in a vein, consider the possibility of an underlying malignancy. And so this was a fairly profound observation. Ironically, he foretold his own clinical outcome. He developed a left upper extremity blood clot about a year and a half later. And there's some question whether he had pancreatic cancer or gastric cancer, but regardless, he died about 6 months after that. And this observation to this date remains true. I'll show you some background around this. Now, platelets are obviously known for having a role in forming blood clots, but now it's increasingly clear that they do far more than that. They are now recognized as large reservoirs for angiogenic and oncogenic growth factors. By the way, thrombocytosis by the NHLBI is defined as a platelet count of greater than 450,000 per microliter, and that's a definition that I'll use during the talk. Significance of paraneoplastic thrombocytosis was really not as well understood, but now platelets are well recognized to play roles in tumor growth, inflammation, and this is a rapidly growing area related to immunity. Dr. Mani from here has also done some really elegant work in this regard. This is an EM image showing you that platelets are incredibly rich in what they contain. So the alpha granules pointed to by this orange arrow contain a lot of pro-angiogenic factors. They do contain some anti-angiogenic factors as well. Before Judah Folkman died, this was one of the areas that he was really trying to push to try to understand how these growth factors get packaged and released from these kind of granules. They also contain dense granules, as shown by this blue arrow, that contain some inflammatory substances, and then also have they also have some lysosomal contents, including proteinases and glycosidases. So let's talk a bit about clinical relevance of high platelet counts, and then I'll get into some of the mechanisms. This is one of the really intriguing studies that I would encourage you to take a look at if you're not familiar with this, where Prandoni reported in New England Journal in the early 1990s. They looked at individuals who had either a secondary thrombosis, secondary meaning that there is a known underlying cause for a blood clot development, such as protein C or protein S deficiency, or they had looked at individuals who developed an idiopathic thrombosis, either a first event or recurrent idiopathic thrombosis. Idiopathic meaning that there's no obvious underlying cause for the blood clot. And they tracked these individuals and found that within a 2-year period, it's really incredible that those who have recurrent blood clots, almost 1/5 of those individuals were diagnosed with an underlying malignancy. Those who had a known reason for a blood clot really had fairly low rates of being diagnosed with cancer. Another study that's fascinating is that if you look at individuals who just walk off the street and have a CBC drawn for whatever reason, so in this study they had about 14,000 unselected individuals. 82 out of those were diagnosed with thrombocytosis. Almost 40% had an underlying malignancy that was recognized once these individuals were worked up. Again, the clinical relevance for some of this was not as well understood, but there aren't too many markers that are this good where almost 40% of patients end up having an underlying malignancy. So the point being that does everyone have cancer who have high platelet counts? Of course not. But don't forget about the possibility of an underlying malignancy that should be considered. So in the context of ovarian cancer, there really wasn't a whole lot of data. So one of our fellows, clinical fellows, Becky Stone, who's now at Johns Hopkins as a faculty member, she looked at about 600 patients with epithelial ovarian cancer and found that about 1/3, so about 31%, had thrombocytosis. And again, anecdotally, we kept seeing a lot of patients who had high platelet counts. And she also found that about almost 1/4 of these women will be diagnosed with a blood clot at some point in their care. This is not all at primary presentation, but at some point in their care. Nicole Fleming, one of our faculty members here as a part of our Moonshot effort, has recently found that about 12% of women who undergo neoadjuvant therapy at primary presentation or during therapy will have a blood clot. Now, with thrombocytosis, patients tend to do poorly. Their progression-free survival and their overall survival is poor compared to those who have normal platelet counts. Now, as I was getting ready for this talk, actually, since we reported that in 2012, there have been a number of other studies, whether you look at Mayo, Europe, and other institutions in the United States. It's a very consistent finding that with thrombocytosis, you tend to see poor overall survival, increased mortality, and so on. Now, there are specific histotypes of ovarian cancer that have even a more profound relationship with regard to thromboembolism. One of our other prior trainees, Koji Matsuo, looked at ovarian clear cell carcinoma and showed that those who have advanced clear cell ovarian cancer, almost 50% of these women will be diagnosed with a blood clot at some point in their care. That's just a huge incidence. And, again, many of you will recognize that these are not benign events. This kind of thromboembolism has real morbidity and can also have mortality associated with it. So obviously, we need to do something about this. We brought this back to the lab to try to understand the mechanisms of thrombocytosis. There was really not much data around how thrombocytosis evolves. We looked at a number of mouse models of ovarian cancer and found that as tumors become established and get bigger, you see anywhere from about a 30% increase to near doubling of platelet counts in mice. And there's a very direct correlation with the amount of tumor weight and the extent of tumor nodules that you see in these animals. And if you look at syngeneic animals that are in a C57 immune-competent background, similarly, you see elevation of platelet counts. If you look at even other tumor types, such as endometrial cancer or pancreatic xenografts, you similarly see increased platelet counts. This is a non-metastatic model of breast cancer, so you see slight elevations in this setting, not as profound. We kept observing that the spleens in mice get bigger as tumors get established. So Becky looked at the bone marrow, but also the spleens. By the way, spleen is a megakaryopoietic organ in mice, and found that as tumors become established over time, that the megakaryocyte load becomes quite high, both in the bone marrow and spleen. Now, in a human setting, we don't routinely do bone marrow biopsies in our patients, but working with our pathology colleagues, we did find several patients who, prior to any treatment, had a bone marrow biopsy. We found that those who had thrombocytosis tended to have megakaryocyte hyperplasia, and those who had normal or low platelet counts had relatively normal megakaryocytes, or even perhaps somewhat of hypoplasia. Now, just to maintain homeostasis, this is an incredibly dynamic system, so you need to produce about 100 billion platelets on a daily basis. And platelets in an in vivo setting live for about 7 days. If you take platelets out and transfuse them into patients, they have a lifespan of about 3, perhaps 4 days at most. These are a lot of the factors that are related to different steps in megakaryocyte and platelet development. We really didn't know what could be contributing to this paraneoplastic thrombocytosis. So Becky took about plasma samples from about 150 women with ovarian cancer and measured a lot of these factors that are thought to play a role in platelet development. TPO was obviously expected because that's known to play a role in platelet biology, but actually the top hit that was related to platelet counts was interleukin-6, and that we were not expecting. So we went further to try to dissect the mechanisms. First, at a correlative level, we found that those patients who had thrombocytosis tended to have substantially higher circulating IL-6 and thrombopoietin levels. So our hypothesis was that tumor-derived interleukin-6 would stimulate TPO production from the liver, resulting in megakaryopoiesis and thrombocytosis. So we looked at the livers from animals as the tumors become established at a message level and saw significant elevations in TPO levels. Now, in a human setting, obviously that's not easy, but we do partial hepatic resections in some of our patients who have limited number of metastases to the liver. So Becky dissected out the normal liver part. She didn't take the tumor. She took the normal liver tissue rim around the tumor and looked again at a TPO message level in these patient samples and found that there was a significant correlation with TPO mRNA levels and platelet counts in these patients. Now, we needed definitive proof of whether this hypothesis was correct or whether this was wrong. I was at a seminar where Angela Drew from Cincinnati was in the audience, and she had created a number of IL-6 or IL-6 receptor knockout mice, and she was kind enough to share a mouse model that lacks IL-6 receptor only in the liver, no other place. So in this mouse model, if you look at wild-type animals, again, the platelet counts go up, so this is a syngeneic model. But if you look in the IL-6 receptor null model, the platelet counts don't rise in this setting. And if you look at the plasma TPO levels, similarly, in the null mouse model, they don't rise, suggesting that this indeed may be the pathway relevant to thrombocytosis. We then took a neutralizing antibody against IL-6 called cetuximab and treated ovarian cancer mouse models. So these are hybrid models, meaning that these are human ovarian cancer cells injected into nude mice. And we treated them with this antibody. And again, it impaired tumor growth, but our main interest was to see whether or not platelet counts are impaired. And indeed, when you treat with the IL-6 neutralizing antibody, platelet counts don't really rise in these animals. Now, this is all well and good in a preclinical setting. We really had to look whether this was true in a human setting. Again, I was at a seminar where Ian McNeish and Frank Bockwell from University of London happened to be there. They had just concluded a clinical trial, a phase 2 clinical trial, with the same exact antibody, with cetuximab, in women with ovarian cancer. And they treated 18 patients with platinum-resistant ovarian cancer. And they were not thinking about platelets, but I requested if they could go back and look at platelet counts and send those to us. And we were really quite amazed that within a 3-week period of time, the platelet counts largely normalized after starting the IL-6 antibody, again suggesting that this is an important axis in this context. So for this part, the mechanism that we think is operative is that tumor-derived IL-6 stimulates the liver to produce TPO, which results in megakaryopoiesis, resulting in thrombocytosis. It's likely that there will be other factors that play a role in thrombocytosis as well that would be important to understand, but IL-6 clearly seems to play an important role in this biology. I wanted to show you a couple of other things related to platelets in cancer patients. We collaborated with Hua Chu to look at the platelet ultrastructure. Hua Chu was at Baylor at the time. Now he's at Stanford. He's one of the world's foremost authorities on cryo-electron tomography. This is an annotated image of a platelet in a resting state that has just beautiful architecture with a rim of microtubules and then alpha and dense granules and some mitochondria. In an activated state, the whole ultrastructure gets incredibly reorganized, as you see with the microtubules, but the microtubule content is completely intact in this setting. The reason I want to show you this is that we, with Hua Chu, looked at a number of individuals where we looked at resting platelets from those who don't have any type of a disorder, or looked at patients with benign adnexal disease or those with malignant disease, and then annotated these and find that, again, the resting platelet from a normal individual has completely normal architecture. Those with a benign disease also have very similar platelet ultrastructure, but we were just fascinated to see that those who have malignant disease have markedly reduced microtubules in the platelets. There are also defects in mitochondrial content, and there are these vacuole-appearing structures that may very well be the open canalicular structure in the platelets from patients. This might actually be an opportunity to serve as a biomarker. The trouble at this point is that this is an incredibly labor-intensive process to do the cryo-ET and annotate those, but we're hoping that there are other strategies that we can develop around platelets. Now I'm going to shift gears and go over some of the roles of platelets in different aspects of tumor biology. The first example I'll focus on relates to anti-angiogenesis therapy, and bear with me while I set the stage for this particular project. Anti-VEGF therapies for ovarian cancer are FDA approved in 3 main indications, for upfront maintenance and for treatment of platinum-resistant or platinum-sensitive disease. These are pivotal phase 3 clinical trials that led to FDA approvals, and they consistently show a benefit for progression-free survival. Most of these studies don't show a benefit for overall survival. For ovarian cancer, this is not a good endpoint to look at for a variety of reasons. But the one really intriguing thing that we see in the results from these trials, though, is as follows. This was a European trial called ICON 7, where patients received either chemotherapy alone, shown in the blue line here, or they received chemotherapy with bevacizumab followed by a maintenance phase. The red box here shows where the maintenance treatment with bevacizumab stopped. And what you note is that the PFS curves actually collapse. Some clinicians call this a banana effect, meaning that the curves come together at the end, perhaps even cross over. This is a really strange phenomenon. We see this consistently with anti-VEGF therapies in patients with ovarian cancer, colorectal cancer, and even other tumor types. And so I went back and looked at the last 40 plus years of nationally done phase 3 clinical trials that look at just chemotherapy alone. These are just two examples of GOG 111 and a Japanese GOG study. You see the red box again is where the intervention stopped, but the PFS curves don't collapse together. We never see that in chemotherapy alone clinical trials. So there is something unique that we saw, or we continue to see with anti-VEGF therapy trials. There are many questions around this. For today's purposes, the question that we asked was, could there be accelerated tumor growth following cessation of anti-angiogenic therapy? This is a Italian study that came out of Rome by Anna Fagotti's group. They showed that patients who receive bevacizumab as first-line therapy at relapse, their time to progression on second-line chemotherapy tends to be shorter, and if you operate on these patients, the likelihood of achieving complete cytoreductive outcome, or R0, tends to be far lower if they had received bevacizumab as first-line therapy. So that made us wonder, could there be some fundamental reprogramming that occurs in the tumor or tumor microenvironment as a result of such therapy? Monica Hammerly spent about 2 years in our lab. She's a pathologist who came from Germany. She looked at mouse models where we gave either continuous anti-VEGF therapy or we withdrew it after a period of time. By the way, this withdrawal period can be after 7 days, 14 days, 21 days, it doesn't really matter. If you do that, so here, pazopanib is a pan-VEGFR inhibitor. Those who continue with treatment obviously have some benefit from it for a period of time. After that, there are adaptive changes. But those animals where you withdraw the anti-VEGF therapy, the rebound tumor growth is actually greater than as if they had never been treated with anything. This is not unique to a small molecule inhibitor. We saw the same phenomenon with a anti-VEGF antibody as well. We've done a lot of work to understand the adaptive changes in response to anti-VEGF drugs, and there are indeed changes that occur in endothelial cells, pericytes, tumor cells, the immune environment, fibroblasts, etc. But for today's purposes, I'm going to focus on the role of platelets and how that can contribute to some of this biology. Now, we wondered that if platelets can only interact with cancer cells in the circulation, and by the way, ovarian cancer cells are indeed present as CTCs in the circulation, that's a fairly finite ability for them to interact with cancer cells. So we asked the question, can platelets extravasate outside of blood vessels and be present in the microenvironment? At the time that we studied this, we really could find no data on this. So we started to look at this at many different levels. We collaborated with Franziska Gushiken, who had created a PF4 Cre-driven mouse model that had a YFP expressed only in the platelets. We took out the platelets from these animals and infused these into tumor-bearing animals. Now, if you surgically resect a tumor, theoretically, you could cause an artifact by creating bleeding into the tumor. So to address this question well, what we did was actually, prior to killing or sacrificing the animal, we perfused the animal with saline, then perfused the animal with paraformaldehyde, and then resected the tumor. So whatever got into the microenvironment would have had to have gotten there by perfusion. If you do that, Becky found that outside of blood vessels, you actually find quite a few platelets in the tumor microenvironment. Platelets, by the way, are about 2.5 microns in size on average, so they're far smaller than an immune cell. And if you look in the ascites, she captured this beautiful image of a platelet bound to a tumor cell in the ascites from a patient. Back to the anti-angiogenic question, we find that when you withdraw an anti-VEGF drug, paradoxically, the amount of hypoxia actually goes up quite substantially in these tumors. And you see that the blood vessels are much leakier if you look at using dextran perfusion compared to if you continually treat these tumors. Moreover, if you look at the vascular structure, what you find is that the vessel density following rebound compared to controls when you withdraw the therapy is actually greater, and the vessels tend to be poorly covered by pericytes, so they're actually less mature. Now, in the 1960s, we learned that the two most robust activators of platelets are epinephrine and ADP. In this context, we find that under hypoxic environments, ADP levels go up quite substantially compared to a more normal oxygen tension. So with prolonged anti-VEGF therapy or withdrawal, as I demonstrated on the last slide, we know that levels of hypoxia are quite profound in the tumor microenvironment. We looked at this additionally, and again find that the platelet extravasation following withdrawal of an anti-VEGF therapy is much greater. These are not easy images to capture, but just an example EM image of a platelet on its way extravasating into the microenvironment between two endothelial cells. I thought it was just a pretty image to show. Now, if you prevent platelet counts from going up by using an anti-platelet antibody, so here we used a monoclonal antibody targeted to GP1B alpha, we're not trying to cause thrombocytopenia, but rather preventing thrombocytosis. Again, in these animals, if you look at withdrawal of anti-VEGF therapy, the rebound tumor growth is greater than controls. But if you prevent platelet counts from going up by using this APA antibody, you can abrogate that kind of an effect. This antibody, by the way, is really quite specific for platelets. We wanted to make sure, so we looked at platelets that, of course, it recognizes by flow. It does not recognize cancer cells, endothelial cells, pericytes, or many other cell types that we looked at. Now, there was some biology emerging that focal adhesion kinase is thought to play an important role in platelet motility and its movement. So we obtained a mouse model from Ian Hitchcock, who had created a PF4 Cree-driven model that lacks FAC only in the platelet compartment. And many of you will know that FAC is also expressed in endothelial cells and so on. So we wanted to look at this very specifically. In this mouse model, again, if you withdraw the anti-VEGF drug in wild-type animals, you see much greater rebound tumor growth. But if you look at this mouse model that lacks FAC only in the platelet compartment, you don't see this kind of increase in tumor rebound, and you don't see the extent of platelet extravasation in the microenvironment that you see in the other animals. At a translational level, FAC inhibitors have been developed. They are in various stages of clinical development. We looked at these, and in these mouse models where we withdraw the anti-VEGF drug, again, either a small molecule or antibody, but you have a FAC inhibitor on board, you don't see that kind of rebound or accelerated tumor growth. So for this part, the mechanisms that we think are operative is that in response to the profound hypoxia that occurs and increases in ADP levels, you have a less mature vascular bed. You see a marked increase in rebound angiogenesis as well as tumor growth. And if you shut down focal adhesion kinase or block its activation, you see that the vascular bed is maintained in a more mature state, and you prevent the kind of rebound tumor growth that's seen otherwise. Translationally, a FAK inhibitor we feel could be combined either with a anti-VEGF drug, or it could be given even in a maintenance setting after the VEGF-targeted drugs treatment is finished. We're certainly interested in having the ability to do those kinds of clinical trials. The last example work that I'll share with you relates to the role of platelets in preventing anoicus and facilitating metastasis. The role of platelets in stimulating metastasis is really it's not a new concept. That's, again, been known at this point for almost 50 years, where in the 1950s and 60s, scientists did some experiments with platelets and injecting cancer cells that would metastasize to the lungs. Anoicus, as most of you know, stands for homelessness, and it's a concept that cancer cells have to be able to overcome in order to survive the metastatic process. And we wondered to what extent platelets could actually provide a survival advantage for cancer cells. A few years ago, Miriam Labelle in Richard Hines' lab from MIT published this really elegant study that showed that platelet-derived TGF-beta promotes EMT and facilitates cancer cell extravasation and establishment of metastasis. Do you see, most CTCs are not present in isolation in the circulation. Most cancer cells in the circulation are actually coated quite heavily with platelets. It is now known that about 90% of TGF-beta in the circulation is really carried by platelets, so they play a very important role in this context. Miriam didn't really look at cancer cell survival in this particular study, and that's what we wondered about. So Monica Hammerly, again, also did this particular project. She looked at the same antibody that I mentioned earlier, the GP1B alpha antibody, and looked at ovarian cancer as well as colon cancer models where the tumor cells for this colon model are injected into the spleen and allowed to metastasize to the liver. If you block platelet counts, you see a much greater extent of anoicus in the ascites of animals where the cancer cells are largely suspended. They're not attached to anything. Moreover, the number of metastases is markedly reduced if you block platelet count. In an in vitro environment, if you prevent cancer cells from attaching to the matrix or plastic or whatever surface you're working with, and keep them in a suspended state, with increasing number of platelets, you see greater and greater cancer cell survival. Again, this is not uniform for every single cancer cell, but these are some examples where we do see that kind of an effect occurring for protection of cancer cell survival. We tried to understand the mechanism for this and looked at incubation of platelets with cancer cells, and looked at both protein in both compartments and mRNA in the cancer cells. Platelets, as you know, are anucleate and contain very, very low amount of mRNA. We collaborated with Steve Wong at Methodist, who had created software to be able to look at 2 or 3 compartment settings, where you can look at what factors could be stimulating the receptors on the second cell type and then triggering signaling in the second cell type. So in collaboration with Steve, we discovered that YAP1 was really the dominant pathway that appeared to be activated in cancer cells in response or after incubation with platelets. So Monica, to validate this, looked at a Western blot level and found that YAP phosphorylation levels with incubation with platelets go down quite substantially, which, as many of you will know, is required for it to migrate to the nucleus to set off transcriptional machinery. And indeed, following incubation with platelets, she saw YAP1 migrating to the nucleus. If you silence YAP1 in the cancer cells, you can restore anoicus even when the cancer cells are incubated with platelets. To further look at this, she looked at a cancer cell model that had relatively low levels of YAP, and she looked at three constructs, either wild-type YAP or constitutively active YAP or a signaling defective YAP, and found that the wild-type and the constitutively active YAP indeed protect cancer cells from undergoing anoicus. That's not a surprise. It's known to play a role in cell survival. In a in vivo setting, she saw that the effect was really mostly on the metastatic disease. So these are fully orthotopic tumors where the cancer cells are surgically implanted directly into the ovary, not into the peritoneal cavity. In this setting, the metastatic disease is far greater with YAP activation. The primary tumor was not impacted to the same extent. In collaboration with Gabe Lopez and our RNA Center, we used a DOPC platform to deliver siRNA to silence YAP in the tumors, and find that you can, if you silence YAP, even if you artificially cause thrombocytosis with platelet transfusions, if you silence YAP, you can completely abrogate the increased tumor growth that you see with thrombocytosis. With the DOPC siRNA, indeed, you can reduce YAP1 levels, whether you look at a Western blot or immunohistochemistry level. So the model that we think is operative is that in response to platelets, there is YAP activation in the cancer cell compartment, which goes to the nucleus and triggers activation of machinery that helps to prevent cancer cells from undergoing anoicus. So in summary, we've learned that paraneoplastic thrombocytosis and thromboembolic disease is really a fairly common event in ovarian cancer and in many other cancers. We've learned that extravascular platelets play a very important role in accelerated tumor regrowth following anti-VEGF therapy, and we feel that disrupting platelet-tumor interactions may offer important opportunities for therapy. There are many, many ways of doing this. One example that I'll just show you relates to use of baby aspirin. There's been very consistent data for colorectal cancer that with baby aspirin, there's reduced incidence of colon cancer, and there is also improved outcomes of patients who are on aspirin. For ovarian cancer, that kind of data didn't really exist. One of my collaborators, Shelley Tooroger, when she was at Harvard, she did this analysis of nurses' health data. So this is an epi-based study, and showed that, and the power of her study was that she could look at pre-diagnosis use of aspirin as well as post-diagnosis use. So really compelling data that Shelley showed that patients who have post-diagnosis use of aspirin have a hazard ratio of 0.68. So Again, is this definitive data? Of course not, but it's thought-provoking in the context of using aspirin or perhaps other drugs to disrupt tumor platelet interactions. Since then, Shelley has actually been funded by the DoD to do a prospective study with aspirin in patients with ovarian cancer that's meant to be information or translational rich study. It's amazing that that institutes at NIH at times don't really talk to each other. So after some of our work had been published, I was fortunate to be asked to co-organize a first of its kind meeting between the NHLBI and the NCI a few years ago, and then in 2016 we published a white paper from this along with Nigel Key and many of the other participants. And if you're interested in this area at all, I would encourage you to take a look at this, where we tried to define some of the priority areas moving forward. Moreover, after this meeting, there was a RFA that was generated to fund research. And we think that additional program announcements and so on are likely to come out to stimulate research in this important area. With that, I'll stop. And I already acknowledged the first authors of the studies whose work I showed you, but I want to very much acknowledge my lab members and, importantly, many collaborators, especially Vahid Afshar, who's a benign hematologist here and has been a collaborator now for 10+ years, and then also members of our RNA Center with whom I've had long-term collaborations. Thank you again for allowing me to present today.
Daniel, this was a wonderful presentation, very interesting, and a topic that is not all the time taken into account. And in fact, one interesting question. So one of the attendees saying cancer-induced IL-6 and thrombocytosis and eventually fatal outcome. COVID-19 induced cytokine storm, and then it's a growing evidence for microthrombosis in lung. Have these two scenarios something in common for treatment strategies?
Yeah, it's a great question, and I actually have had several conversations with Vahid Afshar as well as David Mentor here, who's also a platelet expert. That while there may be some overlaps, the two settings are also different in the context that in a tumor setting, the IL-6 levels are elevated in a sustained fashion for a prolonged period of time. Whereas in a COVID environment, the IL-6 is a fairly abrupt or a rapid increase, and it's really part of a cytokine syndrome, and there are many other inflammatory factors that are markedly elevated as well. And so the changes or the clotting that we see, it may not be the same as what we see in a cancer environment. There are likely to be differences. And the perspective that I shared with you today was obviously very platelet dominant. Obviously, the coagulation cascade and other factors are likely also to play a role that we need to understand.
Another question. So one participant, great talk, Anil. Could the reorganization of platelets, microtubules, and granules in ovarian cancer patients be an effect of chemotherapy? Is this reorganization found in chemo-naive patients?
Yeah, great question. So all of those samples or examples I shared with you were at the time of primary diagnosis, so that they had not been exposed to any cytotoxic drug, because we worried about that exact aspect. We have looked at platelets following treatment with paclitaxel, but only to a limited amount, and that is an area we would certainly love to develop further.
Another question. So very nice lecture. It is possible to manipulate genetically platelets to activate the immune system to attack cancer cells? And also, are platelets in the exosome family?
Great questions. So platelets, as I alluded to earlier, come from megakaryocytes, and they're literally sheared off in the blood vessels from what are called tendrils from megakaryocytes. Theoretically, it is possible to engineer megakaryocytes to express things of interest. There are a couple of groups internationally who are trying to develop systems where you can create platelets in an ex vivo setting. Those are still early, but it is an exciting area to follow because, as we constantly get emails right now where our patients need platelet transfusions and so on, if we could create an ex vivo system that could efficiently produce platelets that we could give to our patients as well, it would be an enormous advance, and then it could certainly have implications for tumor biology as well. George, I'm sorry, can you repeat the second part of the question?
What was are platelets in the exosome family? So probably
Yeah.
platelets can be considered a category of exosomes. I try to.
So platelets actually release quite a few exosomes, and we are actually doing some work in that area as well. So yes, absolutely, they release exosomes, and there is some thought that larger extracellular vesicles may even contain components of platelets within them.
Other question: any correlation between platelet count and the response to VEGF therapy in day one?
Again, a very important question. It's the question that I don't have the answer to. Again, Vahid and I have discussed this and are very interested in looking at that question, but it's not something that I can answer today.
Another question, technical. How could we extract platelets to use for in vitro experiments?
Okay. If I could trouble you, just email either me or Vahid, and we can send you those kinds of protocols. Those are nicely established protocols in terms of extracting platelets for use. And you just have to be careful that you don't activate the platelets while you're isolating those. So it's a protocol that we would be delighted to share with anybody who would like.
Another question. Nice talk. What about using BCL-XL inhibitor to reduce platelet, or the dual BCL2, BCL-XL inhibitors to reduce tumors and platelet at the same time?
I think it's an intriguing thought and very much worth considering. Again, it's not something that we have data for in our group at this point, but in my view, that would be worth testing. Yes.
Some cancer patients get platelet transfusions and the platelet counts drop. There is evidence that these transfused platelets could be activated. What are your thoughts about whether these platelets could actually make their disease worse?
It's a complicated question because in cancer patients now, over the many years when I was a student, the thresholds at which we would give platelets were actually higher. But over the years, the thresholds have gone down. In that kind of a setting, if you don't give platelets, then you're sort of stuck because your risk for bleeds becomes substantial, and especially for major bleeds. And those are incredibly sick patients at that point in time. So could transfused platelets in that setting stimulate growth? It's possible, but you're also dealing with an environment or in a setting where those patients are profoundly thrombocytopenic already. And we really try to avoid giving platelet transfusions because after a certain number of transfusions, patients also start to develop antibodies as well. So we really try to keep platelet transfusions to a minimum.
It's a balance between the immediate need and the potential long-term effects. And one question from me. So Anil, what is known about the non-coding RNAs from platelets? Are they useful or they are detrimental for cancer, for tumor cells?
Yeah, great question, George. So there are non-coding RNAs that are unique to platelets, and we think that they may play some role in stimulating endothelial cells and perhaps even either stabilizing vasculature or in stimulating angiogenesis. We don't have a lot of data in that area, and that's certainly something, George, with you, we would certainly love to collaborate and develop further.
So, Anil, thank you for your wonderful talk. It was really very interesting and a lot of questions, a lot of interest. And also I want to thanks to all the participants. Again, without you, these webinars make no sense. And with this said, you can go on the site of MD Anderson for this webinar in order to see all this very interesting presentation. And keep in mind, after rainy days come sunny days, so we will move forward enjoying science and doing all the best to make discovery for the patient and colleagues. So see you Thursday morning, another great presentation by Jeff Myers, Friday, Manis and I, and with this all the best, take care and see you Thursday. Thank you for being with us to enjoy science.